All right, so the buffers in the body that I mentioned, there's the phosphates, which is what the buffer intracellularly, and then there's the bicarbonate, or the carbon dioxide. And this one in particular has got a special name. This is called the Bohr effect. And so if you notice here, it's not just a single reaction, because carbon dioxide itself is not very water soluble. That's the reason why whenever you open a new can or a new bottle of soda and you hear the psh, because it has carbon dioxide, it's carbonated. It's not, the gas is not very water soluble, so it escapes. Okay, and that's how sodas go flat. However, our body actually uses that to our advantage, in the sense that what happens is it takes the small amount of carbon dioxide that goes into the aqueous form, so it makes it a little bit soluble, but then it immediately reacts with water to form the bicarbonate. The bicarbonate itself then can dissociate to get H plus and HCO minus. So therefore, if you take something in, let's say you had a sub sandwich and you put vinegar on it, then what that's going to do is that's going to force the equilibrium to go backwards. But we have enough of the bicarbonate here to react with a little bit of acid and just ultimately to make more carbon dioxide gas for you to exhale. Likewise, if you become hypoxic, what does hypoxia mean? Hypo means not enough or low. Hyper when you're hyperactive. But hypo and then oxic means there's just not enough oxygen, which is another way of saying there's too much carbon dioxide. And so with that, if you increase the amount of carbon dioxide in your blood, you're actually going to cause the pH of your blood to go up or to go down. It goes down because the equilibrium, the excess of carbon dioxide is going to react with the formed bicarbonate and then ultimately to decompose down to the H plus. And so since you have an increase in H plus, it's a decrease in the body's pH. So that's why you actually can get an acidemia. The pH of your body will actually start to decrease. You get too much acid if you're not getting enough oxygen. Likewise, if you get lots of oxygen, then the pH will go up of your blood, okay, because the fact is it's the exact opposite effect. All right. So the way to select a buffer, which next semester in lab, you'll have to start selecting some buffers, make some buffers, is you want to look at for a couple things. One is a suitable pKa. Like I said, if you're going to be working around in the in vivo pH, around pH 7.4, then you're not going to want to use acetic acid because it's horrible. Whereas if you're wanting to look at something on the way that acids affect, like an enzyme or something, do a pH profile, and you get down to like pH 5, acetic acid is great. And it should also make sense that it shouldn't interfere with whatever assay that you're using. It needs to be a suitable ionic strength, because if you add more of the higher the concentration of the conjugate acid and conjugate base, the higher the buffering capacity. However, it's also going to affect the ionic strength, because these are ions, which is that in of itself can affect other things in vivo and in vitro. Like it affects like the ways that metals bind and things like that. It has to be soluble in whatever solvent that you're using, and you want to make sure that it's non-biological in nature, in the sense that you don't want, especially if you're trying to do this with anything in vivo or with like a cell culture, then you don't want it to where the cells just degrade it, because then it doesn't make sense. These are just a couple that we use in the lab. You may have heard me say heapies. That's that top one. Mops and then pipes. So I would never ask you to memorize that or anything. But I could give you something. Oh, whoops. Maybe I don't have it on this one. I don't have it on the site. But if I give you a list of possible buffers and their pKa's, and I asked which one would be best or better suited, then I would expect you to compare, okay, we want to look in the pH range of 7, so we're going to use a phosphate or heapies, versus a pH 5 is acetic acid, versus pH 10, maybe arginine. Okay? Just briefly I want to cover osmotic pressure. It's very similar to P-inert, if you kind of look, because it's a pressure then it is related to the amounts that you have and then the gas constant temperature. But all it means is the fact that, for example, people actually use the term osmosis incorrectly. It's diffusion based off of solute molecules. And so with respect to water, usually what we're talking about. And so what happens is if you have a semi-permeable membrane to where the water can flow in, there is some force, a pressure that will cause it, so water will flow in. So yeah, I did good. But the solute can't come out. And so it's going to try to make the concentration on the inside and the outside equal, right? Because the solutes, whatever the ions or whatever the proteins or anything like that that's in the, for example, we do this with dialysis bags, dialysis tubing. The proteins and stuff can't get out of the tubing. But what happens is if you take a solution that has a high concentration of protein, you put it in a buffer, the buffer's going to rush into the tubing to try to equilibrate that, and that's due to osmosis. Okay, and there is an amount of pressure that you can put to force it back out, and that's the way that you can measure the osmotic pressure. The reason why that's important in vivo has to do with your cells. Because you can imagine, the cells really, your cells are essentially like little dialysis tubes, in the sense that the cell membrane itself is semi-permeable. Certain things can come in and out, like water, but a lot of things can't, unless we have transporters and things that can help, but there's just not. And so if you're in an isotonic solution to where the relative concentrations of the solutes on the inside are equal to the outside, there's no net water movement. But the problem that you get, or the problem that can happen, is if it becomes like, say for example, the solution is hypotonic. So hypotonic means that there's lower amounts of the solute on the outside. So therefore, the water's going to come on and move in. The cells can swell up, and then to try to get the concentration of the solutes to be relatively equal on the inside, and it can cause the cells to burst. Likewise, or on the other hand, if it's hypertonic, where you've got too much of the solute on the outside, then the water from inside the cell is going to go rushing out to try to make it equal, and then it's going to become crenated. It's going to shrink, okay, shrivel up. Okay, and so that's why it's really important that it's in an isotonic type of solution. You keep that kind of thing in mind whenever you're working in the lab as well. Are there any questions on that? That should really be a review from Gen Chem and from general biology. On your own, just go ahead and look over ionic bonds and what ionic bonds are, van der Waals, that's hydrophobic, type of interactions and things like that from Gen Chem. Okay. Just the last few minutes, you've still got about 6 or 7 minutes. I want to start chapter 3 because chapters 3 and 4 are really important. And there's a lot of material to be covered. Yes, I know. Once again, make sure you know all 20 naturally occurring amino acids by that date on the calendar. I don't remember what that date exactly is. Right? But this is an amino acid. Let me go back one. All right. So amino acids, all of them have the same, with one exception, all of them have the same generic backbone. Well, even the one that's an exception, the backbone is essentially the same. It's just that the side chain, that's what they call the R group that hangs off, is a little different because it's an intermolecular bond. But you have what's called the C alpha, and all of the naturally occurring ones that are used to make proteins are called alpha amino acids. Because if you remember from organic chemistry, what does alpha mean? It's the carbon next door to ground zero, if you want to think of it. So if it's an alpha amino acid, that means it's next door to the amino group, and it's next door to the carboxylic acid. Okay, and then there's always a hydrogen, at least one hydrogen, and then there's the R group. They all look like that, essentially. It's the R group that's going to change. Okay, it is a Zwitter ion. You remember that from Gym Chem, which means that inside itself, it has a plus end and a minus end. Since the amine group is a base, the carboxylic acid is an acid. Under physiological conditions, free amino acids going to look like that. Because the pKa's, of the two ends are within range of each other. And so it's just easier for the carboxylic acid to give up its hydrogen and the amino group to give it. Okay. And so this is called the unionized form, but in vivo, if it's a free amino acid, under normal conditions, it's going to be this is what our ionic form. These are just some of the general ways of drawing it. You know, the ball stick, the space filling, things like that. I want especially know the basics for all these, but also know why something is different. Whenever I say, oh, there's an exception, for example, the point of proline being an exception in a moment, you should know the reason why. Because it's important. God makes this way for a reason. So the exceptions are really important. All of the amino acids have at least one chiral carbon except for glycine, which is very important. And so what makes a chiral carbon chiral? How many bonds must it make? 4. And what must be special about those 4 bonds? They all are different. And so glycine is the only one that's not chiral because its R group is a hydrogen. So there's 2 hydrogens, and so therefore there's an internal plane of symmetry, and it's achiral. It's the only one that's achiral. All the other 19 are all chiral. Okay? And so there's the D form, and then there's the L form. If you remember from organic chemistry, this is dextrorotary, which means you stick it in the colorimeter, it's going to rotate the light, polarized right. The light to the right. That's a mouthful. Then there's the L, the levo-rotary, which means you stick it in the colorimeter in the pure form, that it's going to rotate at the same number of degrees, but to the left. Right. The vast majority of those that are present in proteins are in the L. And I saw some evolutionary biologists many years ago back in medical school that gave their reasons why he thought that naturally had evolved to the L, and I don't remember why. I thought it was a really boring lecture. So, but they do have hypotheses and reasons why. Now there are a few times that's important in your body whenever it wants a D amino acid, but usually almost all of the enzymes that work on the amino acids work on the L form. Okay, and so it's almost like they all have gloves that are only the left-handed gloves. So, that one example is racemase, which actually converts, especially proline, from D to L back and forth. But once again, we'll cover that whenever we get to it. And so we can do our Fischer projections just like we do in organic chemistry, which those of you who are currently in organic chemistry, we do this in chapter 7, I think it is, probably in October, so you'll be a little bit ahead of the line. They are all called D and L based off of glyceraldehyde. Okay, and if you remember glyceraldehyde from organic chem 2, it is a sugar, but it's the simplest of the triose sugars. Okay, so it's got 3 carbons. That's what glycerol backbone is. And so by the end of this semester, and definitely by the end of next semester, you're going to know glycerol. Glycerol looks like this. And of course, these would be all hydrogens, 4 bonds each. That's glycerol. Glyceraldehyde is when you take the end and you turn it to an aldehyde. So that's glyceraldehyde. Okay. And so whenever you make glyceraldehydes, Fischer projections, you have the 2 possibilities of either D or the L, and that's the same thing. That's where they get also the D and the L from with respect to the amino acids. Now there are 2 amino acids that actually have more than 1 chiral carbon. Okay, and we'll get to those as we go along. All right, so we're going to actually end here before we actually get into all of the amino acids and start focusing on them. So I'll go ahead and I'll end class with prayer.